We started at 8. Where have you been?
Driving.
No, I'm just kidding. I'm just kidding. I'm just playing with you, not really. See, aren't you glad you weren't that late? Yeah. Okay, here we go. We're on basic cell structure. Okay, here we go. How many of you remember micro? Oh yeah, I thought so. Okay, here we go. Pronode. You do. Okay, can you remember that? Pronode. You do. Okay? All right, karyo. Anytime you see karyo, it means nucleus. Okay? So, pro-no, what's it tell you?
No nucleus.
No nucleus. No cell, no membrane-bound organelles. Okay? It doesn't have a nucleus, mitochondria, anything with a bilipid layer except for a cell membrane, it doesn't have it. Okay? Everybody cool with that? Okay, I'm going to make it real simple for you. Now, I'm going to be covering a couple competencies at once, from the back here and up front. Okay, so here we go. Let's draw this, baby. Let's just do a bacilli. Y'all ready? Y'all remember bacilli? Bacilli are rod-shaped. Okay, good. All right, now, these guys, prokaryotic, okay, does not have a nucleus, but they do have DNA molecule. So I'm going to make the, see how I'm making my DNA molecule? I'm coiling it out. But it has a nucleoid region. It's not a nucleus. And what do you notice the shape of the DNA? You remember it's in a loop. Okay, yep, it's all double helix. You want me to enlarge that for you, honey? I'm at that age too, don't worry. You just tell me. Is that better? There you go. Because y'all wrinkling your procerus, so that's the muscle between your eyebrows. Okay. So when you're teaching and people go, oh, we're wrinkling that procerus. It's either that or there's methane in the room. Okay. Yeah. All right. I don't think they'll ask you this, okay, but the area which the DNA loop is found is called the nucleoid region, but I don't think they'll ask you that, okay? All right, the DNA of a prokaryotic is in a single loop, okay? It's looped. It does have some extra little DNAs right here. See them? Okay, and they're called plasmids. Now this will help you with some of your competencies in the fact that plasmids, they're also DNA. Boy, these pens are no, they must get these cheap. Okay. All right, so these plasmids have DNA in them also, okay, and these are the ones we remove from a bacteria, and we can insert a human gene, such as a gene for insulin or a gene for growth hormone, and then we can get the bacteria, we can put it back into the bacteria, and the bacteria will make that protein for us. It'll make insulin or human growth hormone, and it's a waste product, so it just kicks it out. So it's better than using pig or beef. So we know that as biogenetic engineering. It's exploiting bacteria. That's all it is. Okay? But see, when you get towards the back, it's in the part of the genetic competency, it's going to ask you about that. There you go. That's a great example. Okay, here's another cool thing I can do. I can take this right here, this DNA out of this bacteria. I can put, let's say I put a bat gene in it. Okay, and then I take this plasmid and I put it in the plant cell of a cotton plant. Okay, now this is just extra DNA for the cell, the plant cell. It is insignificant, it's never going to be used. But if anybody steals that cotton plant, see, I've patented that cotton plant. And maybe it's good for resisting drought. Now, if somebody steals that, I can genetically track that plant. See how that works? That's pretty cool, isn't it? Yeah. Now, we use this big in industry. For example, there are places in this country in which large corporations encourage local farmers to grow a specific type of corn or soybean or peanut. Y'all with me? Okay, good. I'm trying to be politically correct so I don't have anybody knocking on my door in black suits, okay? If that farmer doesn't want to grow that specific corn, soybean, or peanut and uses a different one, how would that corporation ever know? That's right. They have a genetic makeup of that plant and they would know. Does that make sense? Yeah. And then people visit them and try to encourage them to grow. Yeah. Okay, so that's a big thing. Okay. All right, but I want to show you how biogenetic engineering, how easy is that? Yeah, it's pretty cool. We just splice that sucker. Okay, so we have DNA. We do have a cell membrane bilipid layer. Do y'all remember bilipid layers? Sort of, kind of, maybe? Oh, look at that no. Yeah, I saw that no. It's okay. Okay, we'll get it. All right, and then everybody got that out. Ribosomes. Now, those of you that remember all this, like Casey's already been teaching it, you be patient because we have all levels in here. We have people that are like, wow, that file has not been opened in so long that their parietal lobe is still searching for it. Okay? And then there's some of you that, you've taught this. Maybe it's part of your health, science. But just be patient with those that are going on blank. Okay, ribosomes. Anytime we see ribosomes, what do we know? Protein makers, okay? So these are protein factories. All right, now, the cytoplasm of this bacteria has enzymes in it. So even though we don't have membrane-bound organelles, we still carry on respiration. We still digest food. We still make waste products. Okay, we still do all those things. But we just don't have the exclusive parts. Is everybody good? Mm-hmm. All right. Now, we all know when bacteria makes a waste product, it could be toxic, right? Okay, and so that's where food poisonings come from. In the ER, a good ER doctor that really was good at micro, when they smell a wound or an infection, they can actually smell that wound and kind of have an idea of what bacteria, especially if it's Pseudomonas, okay? They'll know what that is, all right, because there's a specific smell with it, okay?
Burn unit.
Huh?
Burn unit.
Yes, burn unit, that's right. And it's green. So does this help everybody? Okay, most important. What is a prokaryotic cell made out of? Cell membrane, DNA, ribosomes, and a cell wall. Not, though, like a cell wall of a plant. This cell wall is made out of proteins and lipids. Okay? Sometimes this wall is called a capsule. Okay, we can stain this. If we stain it and it doesn't have a nice slimy coat on it, it would be a gram-negative, it would be purple. If it has an extra slimy coat on it, we say it's gram-positive. It's harder to kill. It's harder for antibiotics to get to. Harder for your white blood cells to kill. But they have weapons. Okay? All right, so our shapes here are round, for what? Cockeye, bacilli, and spirilla. Y'all remember them? Okay. Yeah, that was good. I'm about to fire this pin. Okay? All right. Fun things about cockeye to help you. If it's in a line, if it's a strip of cockeye, is that a better one? You are a great assistant.
I try.
If cockeye is in a line, the strip of it, it's called strip. So streptococci is a what? A chain. Okay? If it's clustered together like we are in a faculty meeting, it's called a staff. Get it? Staff meetings, staphylococci. All right, so if it's a cluster of grapes, you've got staph. If it's a strip, you got strep. Cool? It's an easy way to remember that. Okay? All right, bacilli is probably the most common we see it around as far as being symbiotic with us. You'll know what symbiotic means. Both win. Okay, now that's a relationship, so I'm going back into competencies towards the end. But we have, and this is a great example, the symbiotic relationship in our gut, in our nasal cavity, and ladies in our vaginal canals with bacteria. Okay? And the bacteria, which we know in our gut is either methanogens, get it, or E. coli, right? And they're your friends. They're a symbiotic relationship. Symbiotic for the fact that we give them a home to live in. And they, of course, bless us with vitamin K and a sense of humor, right? Yeah, and it's so fun to teach students that flatulation isn't something you did. You did not make that. Yeah, you didn't make that. Yeah, it was made by the bacteria, and your peristaltic just pushed it out of there. All right, now what happens when an organism dies? The relationship changes. Now, this bacteria that was once symbiotic now becomes a decomposer. Isn't that interesting? So it begins to decompose the body from inside out. We've all seen animals like roadkill in which it begins to decompose it. Now the gas is trapped and they bloat. Right? That makes sense? Yeah. Okay. So that's pretty cool, right? Okay. So what happens if the animal or the organism is alive and there's a puncture or a tear in the gut and the bacteria now finds a new home outside the intestines?
In the wrong place at the wrong time.
That's right. Now it becomes a parasitic situation. So now what's something that was once our friend in a symbiotic relationship now becomes what? Parasitic. Now becomes our enemy, and we can have peritonitis. See? Okay, so I'm just showing you relationships with bacteria. Now we have a wonderful relationship with them. For example, in our sinus cavities, the bacilli that live there are symbiotic for us, and they do this. See them? They take up space. And as long as we keep them moist, they're there for us. But in the wintertime, what does the heater do? It dries it out. So they have a tendency to die off. And those little patches that are left exposed allow parasitic bacteria to begin to what? Take a little hold. Especially when they begin to dry out, your sinuses swell a little bit, and then lack of air, and that kind of makes it a little clear yellow mucus. And that is the wonderful culture for a sinus infection to grow. See how neat? Okay, so it's just you're showing students, and you're getting that mindset of how bacteria has that kind of a love-hate relationship with it, okay? It is imperative that bacteria is a part of an ecosystem. It's in the soil, okay? It changes the nitrogen in the air, okay? How much is nitrogen in the air? Y'all know? 78%. 78%. Okay, I'm glad you knew that because that's part of the junior high curriculum. Okay? 78%, but we can't use it. All right, now think about it. What do we need nitrogen for? DNA. Because what's it made out of? A nitrogen base. Yeah. Adenine, thymine, cytosine, and guanine are all nitrogenous bases. RNA. Protein. What is protein made out of? Amino acids. What's on the amino acid? An amino group. Right. Yep. Come on in here. We started at 8. See, yeah, you're over here. You're not very late, just 52 minutes. So. I harass you if you're late, by the way. Okay, so it's an initiation. So, remember how amino groups have the NH2? So once you metabolize the protein, you make ammonia, and that's what makes your urine odoriferous, right? Okay. How many of you have pet cats? Very good. Okay. Yeah, we clean that box every day. Every day, that's right. Because they're carnivorous, they metabolize protein, so they have a lot of what in their urine? Ammonia. Okay, so nitrogen is important to us. Okay, so the bacteria in the soil and in a special plant group called legumes. Y'all are familiar with legumes, the pea family. We have mesquite around here, yes? Very good. Yes, lots of locust trees make the long beans. Desert willows. Of course, alfalfa, sweet yellow clover, any kind of clover. Legume is a big group here, but on their roots, they have these nodules. And in them live a bacteria. And guess what they do with the nitrogen? They turn nitrogen into nitrites and nitrates. And then plants take those and make DNA and RNA out of them. We eat the plant. And we take the RNA and DNA from that plant, from that spinach that you just ate, which was once spinach DNA and RNA, and we break it up into pieces and make it carry DNA and RNA, because you really are what you eat. I want your students to be like, what? It's true. You're made out of cow DNA and chicken DNA and spinach DNA and apple DNA, because you are taking molecules that other organisms have put together, digested them, and then reorganizing them into you. Okay, so nitrogen. So bacteria, I'm trying to get y'all to be biology minded of how this all goes together. You see how it works? Okay, the kingdoms that this guy belongs to are going to be archaeobacteria. You're right, this one's better. And eubacteria. Now this used to be one kingdom. This used to be one kingdom, now it's two. All right? Okay, now y'all see that U again, right? U actually means true. These are the bacteria you're familiar with. Okay, archaeobacteria is bacteria that lives in severe ecosystems, like the vent of a volcano. Does that make sense? Salt Lake, up in Utah, the Salt Lake. Okay, so just remember, archaeobacteria are just severe bacteria, okay? All right, back there, we're right here. Right on the bottom of this page. Okay. All right, is everybody good? What you're seeing me do in the short amount of time is I'm taking part of these competencies in the cell and I'm tying it to the competencies in the back. Okay, who's who? What's up? Didn't know we started at 10.
No, it's not my party.
It's not your party?
It was Laura's issue.
I started the
It was
party at
my party.
7:45. Yeah. All right, here we go. Now let's do eukaryotic cells. Now here, everybody, mentally know this. That means all the other kingdoms are eukaryotic. That's it. That's easy. Okay. So it means protista, fungi. Y'all love fungus, right? You should. You're all fungi and fungals, right? I know. I love that joke. Yeah. Plant and animal. All of them are now going to be eukaryotic. So see how easy that was for your kingdoms? Okay. How's it going, Miss Debbie?
Good.
All right. So we all familiar with this one, yes? Okay. Let me get my arm out here. There we go. Okay. So what do we see? Cell membrane. Okay. And we see a true what? Nucleus. Okay, we see a true nucleus. All right, and what do we see in the nucleus? DNA. Now, let's go to the world of eukaryotic cells. What is DNA to a eukaryotic cell?
Just back to bacteria real quick.
Uh-huh.
If there are no mitochondria membrane bound respiration, where does respiration?
It happens in the cytoplasm. Mm-hmm. Yeah, and all the enzymes for that is in the cytoplasm.
Okay.
Yeah, and keep in mind, they don't really do much They don't do a big Krebs or anything like that. They just are basically lightly aerobic or anaerobic. And it's real easy. They don't do anything elaborate. Okay? All right, now DNA to a eukaryotic cell is the blueprint, right? It has the instructions for the cell. Everybody remembers that, right? Ooh, yes, remember that?
Say that again.
It is the blueprint of the cell, right? Yes, and write as many notes. Remember, we have such a different level in here. Some of you are like, this is a review, and some of you are like, oh, okay. So remember, it's the blueprint, okay? Now, if you're a single cell, you're a protista, like a little paramecium. Looks like a footprint with hair, okay? Amoeba, which we know, you always talk about amoebas because white blood cells move like them with that cytoplasmic streaming, all right? Okay, all right. Euglenas. Get pond water, you're going to get tons of protista, right? Okay, so if you're a single cell, that's just a blueprint of the cell. But if you're multicellular, such as fungi, plants, and animals, now watch this. It's the blueprint of the entire organism. Okay, now keep that in mind, because I'm heading to genetics with that. It's the blueprint for what? The whole organism. So every single cell has the complete instructions for the entire organism. Doesn't mean it's going to use all of those instructions. So here's a great example to wrap your mind around. I go to Sears and I get a cool entertainment center. Okay, it's the kind you have to put together. All right? Okay. We all know that. When you first get out of college, that's your first furniture, right? Okay. And then when you start raising kids, that's your furniture because who cares if it gets broken, right? Okay. You can't have anything nice until they're in college, all right? It's a true story. Huh? They're right on it. Yeah. Okay. So I get it, and I open up the instructions, which is the DNA for the entertainment center, right? Okay. It has option A. Option A, I can set the shelves up this way and put the TV to the right. Option B, I can put shelves up here and put the TV in the middle. Option C, I can put the TV in the middle and arrange for the shelves to go over it. So I have three what? Genes. See it? So my instruction booklet has 3 genes. It has all the instructions, but I'm only going to use what? 1. I'm only going to use 1 gene. All right, so if I'm a skin cell, okay, I'm a squamous cell, I have all 46 chromosomes of what? All of them. But I'm only going to use the genes to be a what? Get it? To be a skin cell. Okay, does that help you? That's why, now watch this, this is part of your competencies. I can take what is called a stem cell and I can encourage it with specific type of hormones to become what? That's right. For example, now y'all, this is in your competencies, okay? I'm going to give you this example. The latest and coolest thing we're doing with kidneys is we take rat kidneys, we take the stem cells from those guys, okay? Okay, and we can take and build a new kidney with it. All right, so we take the scaffolding of an old kidney and we can take the stem cells from that rat kidney and put it on the scaffolding and it will what? It'll build it. So what are we thinking? Take a human kidney, just the scaffolding of it. I can take the stem cell, let's say of Chris here, okay, and I can put it on that scaffolding and the gene will be turned on to be a kidney and it will make him a new what? Kidney. So if he needs a kidney transplant, we can actually grow a kidney just for him. You see, we're not there yet, but that's the mindset, because we tried to get it where they would build their own scaffolding and that just didn't work. Okay? So that's the kind of neat thing. It's what we're turning genes on and off. Okay, so we're going to get there. All right, DNA. Put a 1 by